Hi everyone, welcome to our discussion on Activity 5. Activity 5 is all about examination of peripheral blood films. Our peripheral blood films are one of the important laboratory procedures inside the hematology laboratory because in cases wherein our machines um, will break down in the middle of processing a specimen, we can have a backup procedure in the form of your peripheral blood films. If you want to do the leukocyte differential count, your peripheral blood film is a very effective way of performing a manual count of the different white blood cells in your patient sample. Okay, and also if you want to perform platelet estimates, WBC estimates, you can also make use of your peripheral blood film. So what we have here is our unstained peripheral blood film, and we also have the stained peripheral blood film. As we go on with our lecture for this topic, you would be able to know what are the different characteristics of your well-prepared blood smear or blood film and how to stain and examine the different cells in our sample using our peripheral blood film. In our next slide here, we can see the different purpose of staining or why do we need to stain our peripheral blood films. Number one, to simply make cells visible. Okay? To be able to distinguish or identify our cells under the microscope, the stain imparts color to our cells so that we can appreciate their forms under the microscope or when we look at them under the microscope. Number two, to evaluate morphological details of blood cells. So when we say morphological details, that includes the form of the nucleus of your cell and also to see if there are granules present inside your white blood cells. So that is the reason why we have to stain our peripheral blood films. And lastly, to check for existing pathological conditions. So basically, our cells, they also tell the condition of the patient by looking at the form of the cell. If there are some inclusions present in your cell, okay, we can see them, these inclusions, because these cells are already stained. So those are the different purposes of staining. For the staining components used for staining our peripheral blood smears, we make use of four solutions. So you have your methanol, your eosine, methylene blue, and finally you have your buffer. So for the function of each solution, we start off with our methanol. So our methanol, it's a fixative solution. It's the simplest of all the alcohols and it's the commonly used or traditionally used fixative for peripheral blood smear. So the purpose of this fixative is that it preserves the overall morphology of your cell, including the nucleus and the cytoplasm. Then next, we have our eosine. Eosine, it's an acidic dye. If you notice the color of your eosine, it's bright orange in color. So it's an acidic dye and it imparts color or stain to the basic or alkaline portion of the cell, and that portion is your cytoplasm. The principle of our staining here is that opposites attract, meaning to say that if your stain is an acidic stain, it stains the basic portion of the cell. Or if your stain is a basic stain, it gives color to the acidic portion of the cell. So moving on, our next solution here is our methylene blue. So our methylene blue is a basic stain. So when what we mentioned earlier that our basic stain, it gives color to the acidic portion of the cell, and the acidic portion of our cell is your nucleus because your nucleus, it contains your DNA and your RNA. So the meaning of our DNA is your deoxyribonucleic acid, or the RNA, the ribonucleic acid. Okay, so that's it. And then the final solution that we have is our buffer. So our buffer, it changes the pH of our solution and it ionizes the reactants to initiate the pH-dependent staining process. So if you notice, our eosine and methylene blue, they have different levels of pH because our eosine is acidic and our methylene blue it's basic. So to balance out these pH levels, we have to make use of our buffer. So those are the different staining components that give color to our peripheral blood smears. Next, we have our Romanovsky stain. So our Romanovsky stain are widely used stains, especially for microscopic examinations that make use of blood or bone marrow films. So some of the group of neutral stains under Romanovsky stain include your Wrights, your Giemsa. We also have your modified Wrights, Giemsa, Leishman, Jenner, and the May-Grunwald. So all of these neutral stains belonging under the Romanovsky stain, they all serve the same purpose, which is to impart color to our cells, to best view them under the microscope, to appreciate their morphological structures and colors under the microscope. In this slide, we can see that there are two staining methods involved in staining our peripheral blood film. So we have the quick dip method and the rack method. So both of these methods, the end product would always be a well-stained peripheral blood smear. But in terms of performing, the easiest and the fastest would be your quick dip method as compared to the rack method. Okay. So what are the staining problems that a medical technologist will encounter when he or she stains her peripheral blood films in the hematology laboratory? So we have two major categories. You have two light stain and a two dark stain. Under the two light stain, these are the possible reasons why you have achieved um, light stain on your peripheral blood smear. It could be that your buffer was too acidic, or the stain was too acidic, or there is insufficient staining, meaning to say that you did not follow the exact time when you perform the quick dip or the rack method, and you also have excessive washing, especially in the last portion of our staining method. After the buffer, you have to wash your slide with tap water or running water. If there is excessive washing, there's a possibility that the stain will be removed, and then finally you have your thin smear. Okay, so if your smear is thin, then of course it would only take a little of your stain, okay, resulting to two light stain on your peripheral blood smear. Then we have the too dark stain, meaning to say that the buffer or the stain that you're using is too alkaline or too basic. And then excessive staining, meaning to say you prolong the time of your staining. You did not follow the exact time for staining your slides, so that would result to a too dark stain. And then we have inadequate washing. Okay, the amount of stain was not properly removed. And lastly, your thick smear. If your peripheral blood film is very thick, of course it would take up so much stain, okay, resulting to too dark stain. For the staining time of our different staining components, we have mentioned earlier that we have our methanol, our AUCin, methylene blue. We also have buffer, and finally tap water. Okay, so for the fixative, which is methanol, the staining time is only 30 seconds. For quick dip, you have to dip the slide 30 times because that signifies that you have achieved a 30-second staining time for methanol. And then for AUCin, which is stain 1, 6 seconds. Methylene blue, 4 seconds. Buffer, our buffer is aged distilled water, that's 45 seconds. And then finally, we rinse our slide with a gentle stream of tap water, that's also 45 seconds, and 3 minutes of air drying. So before you examine your slides under the microscope, you have to properly dry the slide. Okay. Why? Because in the examination of our peripheral blood smears, we have to make use of the oil immersion objective. So we have to make sure that our oil immersion or the oil will not be mixed with excess water as a result of inefficient drying of our slides. So how would you know that you have properly stained your peripheral blood film? You have to look at two areas here. So we have you look at your slide macroscopically and also look at your slide microscopically. So when we look at our slide in a macroscopic view, the color of your slide must be pink to purple. Okay, so if you take a look at our slides here, these are well stained slides or peripheral blood films. The color of the film is violet, okay, or purple. For microscopic aspect of viewing our well-stained peripheral blood films, how would we know that our film is well-stained? We look at our RBCs under the microscope. So if our RBCs have the orange to salmon pink color, okay, so you have achieved the proper staining of your peripheral blood films. And then we also look at our WBCs or white blood cells. The nucleus or the nuclei is purple in color and the cytoplasm is pink to tan, while the granules for granulocytic WBCs, the color of their granules are lilac to violet. Okay, we also have our eosinophils. Okay, our eosinophils are also granulocytic WBCs. The color of their granules are orange. And basophil, the granules of our basophil are dark blue to black. Okay, so when you look at them under the microscope, if your slide is well stained or your peripheral blood film is well stained, you can easily identify your cells under the microscope because yeah, of the color that is or the stain imparted to your cells, okay, during the staining process. Next, we have our zone of morphology. So this is a particular area in your peripheral blood smear where you examine your blood cells. So to be able to distinguish the different WBCs, differentiate them from RBCs and also your platelets, we identify them in the zone of morphology, or the other name for this one is the critical area of assessment. So this is the area where the cells are not too close to each other or not too far from each other. There is enough distance between the cells so that you can easily distinguish them from each other. For the macroscopic examination of our PBS or peripheral blood film, we should always remember that our smear should be well stained and it should have a pink to purple color macroscopically. And the macroscopic view of our slide, it gives the evaluator an indication of what abnormalities or tests that need rechecking. Okay, example, if your smear is bluer than the normal smear, it may be caused by increased plasma proteins, such as seen in conditions like multiple myeloma. Okay. Then, if your smear has a grainy appearance macroscopically, that could mean that there is RBC agglutination, meaning to say that your RBCs are clumped to each other, okay, or they form in groups. Okay, and then if your smear has holes all over it, it could mean that there is increased lipids. Okay, or if if it means or if it's a non-pathological cause, it could be due to technicalities during the smearing and staining process. Okay, so probably your slide or the spreader is not clean or the slide is also not clean, that could create holes in your smear. Our well-stained and well-prepared peripheral blood smear should have three zones or three parts. We have the head, the body, and tail. Okay, so what types of cells are seen in these different areas of our smear? So we have our neutrophils and monocytes. These are white blood cells. They usually predominate in the tail area. Then our red blood cells lie singly, okay, meaning to say there is a space or there's a distance between your red blood cells. Lymphocytes predominate in the body area, and red blood cells overlap to some extent. Especially for parts of your smears that are not in a zone of morphology, most red blood cells really overlap, making it hard for you sometimes to distinguish the morphological appearance of some of your red blood cells and even your white blood cells. So these are the different parts or areas of our well-stained and well-prepared peripheral blood smear. This is our slide. Our slides are usually frosted because that's the area where we label or place the patient's details. And this is our smear. So our smear, it has different areas or parts or zones. We have the head, the body, and the tail. So it is in the tail portion where you can see the zone of morphology. So again, we don't examine our cells using the microscope and look at them if they are seen in the head or body. It's always in the tail, specifically in the zone of morphology. Another illustration here also labels your slides with the different parts. You have your application point, this one. This is the area where you deliver your drop of blood. And then when you push your spreader slide, you create the body of the blood film. And then we have the feathered edge or the tail and your monolayer area. So the monolayer area, this is where you can find your zone of morphology. So the zone of morphology, again, that is the area between the tail and the body, but it's found gearing towards the tail, your zone of morphology or the monolayer area. So don't forget to remember these important parts or area of our well-stained peripheral blood smear. So we're done with the macroscopic examination of our peripheral blood smear. We now go to the microscopic examination of our peripheral blood smear, which is the most interesting part. Okay, so for the microscopic examination of our PBS, we can view our well-prepared smear under the microscope by using the following magnifications. We have the low power objective, the high power objective, and the oil immersion objective. So notice that the different magnifications of your microscope um, are very essential in the identification of the different cells, especially when you go to the critical area of assessment. The most important magnification to use for that is always your oil immersion objective because it has 100 times magnification that makes you distinguish one particular WBC from the other WBCs. And also, you can also look at or see the morphological appearance of your red blood cells and your platelets. Okay, so notice this picture of a blood film. You can see here your different types of WBCs. This one, they are easy to distinguish because they are much bigger compared to your red blood cells. And these are your red blood cells. They are easily noticed because of the central pallor, okay, that they have because of their shape, which is a biconcave disc. And then we have the smallest of the cells. We have your platelets. Okay, so your platelets, they are so small that sometimes you can identify them as dust. Okay, but they are not actually dust. They are just platelets, and they are so small that they are 1/10 the size of your red blood cells. Okay, so those are the things that you can see under the microscope when using the different magnifications of your microscope. When we examine our slide under the microscope using the low power objective, we can actually get an idea of the distribution of our cells in the peripheral blood smear. So the overall smear quality, color, and distribution of cells can be assessed under your low power objective, which has a 10 times magnification. So we look at the feathered edge or the lateral edges of your smear and check for WBC distribution. Remember that our neutrophils and monocytes, they predominate our tail portion of the smear, and then we also have our lymphocytes. They predominate the body portion of our smear, so take note of that. And then our RBC distribution can also be assessed. We try to look at if our RBCs are overlapping or are they forming Rulu formation. So we can check on that by using your low power objective, and that will also give you an idea that your technique for smearing is really effective because if you try to look at your smear under the low power objective and you see that your cells are clumping, then it gives you an idea that the technique you are using is ineffective. Okay? And then we have our monolayer area, that is the area available for suitable examination of our cells. So our monolayer area, that is also our zone of morphology or the critical area of reading or assessment. So again, that is the area where cells are not far from each other or not too close from each other. There is enough distance for you to distinguish one cell from the other. So these are the different parts of our well-prepared blood smear and how they would look like under the microscope. So we have our feathered edge, the monolayer area, and the body area. So to start with, you have your feathered edge, right? So the feathered edge is the tail portion of our peripheral blood smear, the one in color purple. And if you take a look at this portion under the microscope, these are the cells you can see under the microscope. So there is so much space between the cells. The cells are quite far from each other. Okay, and if you can see this field, okay, you can really see so much space, right? So you don't count cells when, or you don't, yeah, distinguish the cells when they are too far from each other. And if you take a look at the morphology of your cells here, it's not the normal morphology of your cell. So some cells would look elongated or too big, too small. Okay, so those are the kinds of cells you can see in the feathered edge or the tail portion of your peripheral blood smear. If you take a look at your body area, this one, the body area is the space between the head and your monolayer area. So for the body area, look at the cells under the microscope. Um, Although there is less space as compared to the feathered edge, the cells here are actually overlapping with each other. So this would give the medical technologists difficult time to distinguish the cells since they are overlapping. So we cannot count in this area or identify the cells in this area. The perfect area to count for or identify our cells is the monolayer area, where you can find the zone of morphology or the critical area of assessment. So if you take a look at your field, okay, when you scan your or view your slides under the microscope, the cells in your monolayer area, the distance between the cells is enough. No, there is not so much space or it's not too crowded and the cells are not overlapping. So, enough lang ang distance between the cells in your monolayer area and it would be easy for you to identify your WBCs, your RBCs, and even your platelets. Okay, so um, although most of the space is taken up, the cells are not overlapping, okay, and there is an equal distance between the cells. This is a realistic photo of your peripheral blood film and its parts when viewed under the microscope. So we have our head here, or this is our application point. So under the microscope, this is how the cells would look like. Your RBCs are overlapping, like, yes, they are overlapping with each other. And then you have here your WBC, but then it's not very clear if what type of WBC is this. So this is what the cells would look like under the microscope if you're going to view the head. If we view the tail portion or the feathered edge of your peripheral blood smear, this is how the cells would look like. The cells are not overlapping, but they are too close to each other. And then there are spaces here, big spaces. So you can see the cells, but there are spaces that should be filled with other cells. Okay, you can identify your WBC right here. If you compare your WBC here with this one looks identifiable as a neutrophil. But if we try to look at your cells under the monolayer or the zone of morphology, look at the cells. They are not overlapping with each other, and there is enough space or enough distance between the cells. You can easily identify the cells. These are your red blood cells, and you have here your WBC or your eosinophil. So see the difference between the different parts of your peripheral blood smear. So this is the head, this is the monolayer, and this is your feathered edge. The zone of morphology, as what we have mentioned earlier, it's the critical area of reading or the optimal assessment area. So it's the area between the thick area where the drop of blood was initially placed or the application point, and the very thin feathered edge. So again, your zone of morphology, it's the monolayer area or the area between the application point and your feathered edge. RBCs are uniformly and singly distributed. That's how your RBCs would look like in the monolayer area, with few touching and overlapping, and have their normal biconcave appearance with a central pallor of 1/3 the diameter of your cell. So if you have noticed earlier that our red blood cells under the microscope, they have a light portion in the middle of the cell. That's the central pallor of your RBC. So one oil immersion field generally contains 200 to 250 red blood cells. So you don't really have to count, guys, no, when you view the red blood cells in the monolayer area. It is expected that in a well-prepared blood smear, there must be 200 to 250 RBC seen in one oil immersion field. We have here another picture of our blood film showing us the cells under the microscope in a monolayer area. So we have here our WBCs, specifically our neutrophils. We also have another type of WBC here, our monocyte. Then we have our red blood cells, okay, the red ones, or the um, orange to salmon pink in color, and our platelets that are so tiny. Okay, you would sometimes see them as dust or microorganisms, but actually they're just platelets. Okay, and then they're so small again that sometimes you neglect them, but they're there. Okay, and they play a vital role in our body. So look at these cells. Although some of the red blood cells here are overlapping, there are still spaces between them that you can distinguish one from each other. Okay, so this is how the monolayer area of your peripheral blood film would look like under the microscope. Next, we have our WBC estimate. Our WBC estimates are performed by using the high power objective of our compound bright field microscope. It has a magnification of 40 times, and WBC estimates are usually performed in the zone of morphology. Okay, so all cell estimates, cell identification, they are performed always in the zone of morphology. So in WBC estimates, what you need to do is you have to count the total number of WBCs in 10 high power fields, and then you get the average of this total number of WBCs you have counted and multiply it by 2000. Okay, so that is the factor that you need to multiply to the average number of WBCs counted in 10 high power fields. So WBC estimates are performed on a routine basis, but in some laboratories, these estimates are only performed when needed, especially if the sample upon feeding into the machine and then the results are quite high, especially for your WBCs, for the different types of WBCs, you really have to perform your WBC estimate. In WBC estimates, when you count the total number of WBCs in 10 high power fields, it won't really matter what type of WBC you were able to identify and count. As long as it's 8 WBC, you have to include it in the count. You only perform the separate identification of your WBC in leukocyte differential counting, but for WBC estimate, it does not matter if the WBC you were able to um, find is a neutrophil, a eosinophil, a basophil, a monocyte, or a lymphocyte. It does not matter as long as it's a WBC, it must be included in the count. Okay? Then finally, we have the examination of our peripheral blood smear using the all immersion objective. The all immersion objective, it has a magnification of 100 times. Okay? It has the highest magnification for most standard microscopes, and this objective or magnification, it's used to perform platelet estimates, red blood cell morphology, and your leukocyte differential count. Okay, we start with our platelet estimate. Our platelet estimate on a right stained wedge smear. So this is also an integral part in peripheral blood smear evaluation. When we perform platelet estimates, we want to verify or confirm the accuracy of the automated platelet count or provide a blood smear quantitative platelet analysis. When your machine registers a very high or a very low platelet count, that's the time you have to perform your platelet estimate. If you want to confirm if really the platelet count of the patient is really high or really low, this is one way of confirming it by doing your platelet estimate. So platelet estimates are routinely performed using your 100 times oil immersion objective, again in an area of your smear we call the zone of morphology. So normally in one oil immersion field we can find seven or platelets ranging from 7 to 21 or 25. Okay, that's the normal number of platelets we can see in one oil immersion field. Under the microscope, using your oil immersion objective, this is how one oil immersion field would look like. So as you can see, we have our white blood cells here. This is your monocyte. Then we have another white blood cell here. This is your neutrophil. And another neutrophil here. Okay, so those are our WBCs. We can also see here our red blood cells. So this is our red blood cells. Your red blood cells actually outnumber your platelets and your WBCs, because in the previous slides it has been mentioned that in one field you can see 200 to 250 RBCs, right? Okay, so these are your RBCs and you can easily identify them because of the presence of the central pallor, okay, due to the biconcave biconcave shape of your red blood cell. And for the platelet estimate, the most important thing here are your platelets. Okay, these are your platelets. They don't really look like dust or microorganisms. They're just so small actually that you sometimes identify them as dust or dirt or artifact or microorganisms. But these are platelets. Okay, they're quite bigger as compared to what we have viewed earlier under the high power objective. Okay, so these are your platelets. So in platelet estimates, you also have to count the total number of platelets per oil immersion field. Okay, what we do in platelet estimate is that we have to count the number of platelets in 10 oil immersion fields. We have to get the total number of platelets in 10 oil immersion fields and then get the average. So that means total number of your counted platelets divided by 10, which corresponds to the 10 oil immersion fields where you counted your platelets. And then we have to multiply the average to 20,000. So that is the factor that you need to remember when you are performing your platelet estimate. So a short recap or a short review, we had earlier our WBC estimate, right? So our WBC estimate, the factor that you need to use to multiply the average number of WBCs counted in 10 high power fields, you have to multiply that to 2,000. So that's the factor for WBC estimate. But for platelet estimate, the factor is different. It's 20,000. Okay, so the reference range for our platelet estimate is this one. I'm sorry, it's already covered with the highlighter. So you have this one for the conventional unit, it's 150,000 to 450,000 cells per cubic millimeter. If you want to convert this one to the SI unit, all you have to do is multiply your conventional unit to 10 to the 6 per liter. Okay, so the SI unit here is 150 to 450 times 10 to the 9th per liter. Okay, so these are the reference range or the normal values for your platelet estimate. After your platelet estimate, another evaluation that we can do using your oil immersion objective is the evaluation on the RBC morphology. So the RBC morphology, it encompasses the size, the color, the shape, and even the inclusions of your red blood cells. So what we need to do is we have to count the number of poikilocytes. Poikilocytes, it's a scientific term. Your poikilocytes, it's a scientific term that means abnormally shaped red blood cells. So we have to count the number of abnormally shaped red blood cells in 10 oil immersion fields and get the average. So after we get the average of the poikilocytes, we then check for the grading of your poikilocytosis. Normally, guys, the shape of our RBC, the normal shape of our RBC is, it should be biconcave shape. Okay, but there are some red blood cells that exhibit abnormal shapes. Okay, take for example this blood film. All of the cells that you can see in this blood film are actually RBCs, but some of them have abnormal shapes, or they are termed as poikilocytes. So an example of that is this one. Okay, so this is an RBC. This is a red blood cell. But if you take a closer look of the cell, the edges of this cell is not smooth. Although it has a central pallor, but it's not smooth. If you try to zoom this picture, the edges or the borders of this red blood cell is actually spiculated, or it has thorns. Okay, so that is considered a poikilocyte. Another one is this one. Okay, so it is not perfectly round, and notice it has no central pallor, and the edges, the lateral edges of the cell are pointed. Okay, so there is a special name for this cell. We call this one the teardrop cell, okay, or the dacryocyte. And then another poikilocyte I saw are this. Okay, the same with the first example I mentioned earlier. The edges of your red blood cell are pointed or spiculated. And this one, the one seen in between these two red blood cells, the RBC is elongated. Okay, so that is an abnormal shape of your red blood cell. So basically, guys, there are so many abnormal RBCs, and they have common names, and they have scientific names that you need to familiarize yourself. Okay, so those are some examples of poikilocytes. So after we get the average number of poikilocytes, we need to grade the degree of poikilocytosis. We have here another blood film under the microscope. So all of these again are RBCs, or red blood cells, but not all of them are showing the normal shape of our RBC, which is a biconcave shape. If you try to study this blood film, you can see here your normal RBC. This is your normal RBC. Okay. And then the rest, or some of them, or most of them, are actually poikilocytes or abnormal red blood cells. For example, we have this one. This is an example of your teardrop cell, or another term for that is your dacryocyte. Okay, dacryocyte, DACRYOCYTE. So again, teardrop cell or your dacryocyte. We also have your this one. This is your acanthocyte or spur cells. Okay, acanthocyte or spur cells. We also have several acanthocytes here, like this one. Okay, acanthocytes or spur cells. What else? We also have your this one. This is an example of your echinocyte or burr cells. Echinocyte or burr cells. Okay. What else we have? Okay, this one. This one, and you also have this one. These are examples of your schistocytes or schizocytes. Schistocytes or schizocytes. If you are unsure of the spellings, you may want to browse your RODAC, okay, your Hematology Clinical Principles and Applications by Bernadette F. Rodak. She has an illustration of the different poikilocytes on page 246. Okay, in the book, you can see an illustration of some of the poikilocytes. Okay, you can see it there. So, guys, again, we have so many or several types of poikilocytes. The one I highlighted in this picture are just some, or they're just few of the many poikilocytes that you need to study and know. Okay? So if we want to determine the degree of poikilocytosis, what we need to do is to, again, count the number of poikilocytes in 10 oil immersion fields. You get the total number of count and then divide it by 10 to get the average. So if your average is within this range from 2 to 10, then you get a grade of 1 plus for the degree of poikilocytosis. Or if the average count of poikilocytes is within 11 to 20, you have a grade of 2 plus. Then if the average is within 21 to 50, that is a 3 plus. And if the average of poikilocytes in 10 oil immersion field is 51 and above, and then so you have a grade or of 4 plus, or the degree of poikilocytosis is considered as 4 plus. So the terms few, moderate, many, and marked may be substituted for the 1 plus 2, 4 plus grading system. So your 1 plus here can be substituted as few, your 2 plus as moderate, 3 plus as many, and 4 plus as marked. Okay, so either way, that would tell you the degree of poikilocytosis in the peripheral blood smear. The last activity that we can perform using the oil immersion objective of our microscope is the leukocyte differential count, but there will be a separate activity for that in activity 9. So for now, let's talk about first about your corrected WBC count. And why do we need to perform this one? So the corrected WBC count will only be performed if during examination of your peripheral blood smear, you can see nucleated RBCs, more than 5 nucleated RBCs in the peripheral blood film. And that's the time you have to perform your corrected white blood cell count. But in our formula here, okay, for the corrected WBC count, we have the WBC count. This one should come from the results generated by your machine. Okay, the results generated by your machine. And then you have to multiply it by 100 and divide the result by the total number of your counted NRBCs plus 100. So the reason why we have to perform the corrected WBC count, it's because when we feed our sample into the hema analyzer, the machine recognizes your NRBCs as white blood cells. Okay, so there will be a false increase of your white blood cells. So in order to correct that, to eliminate the number of counted NRBCs by your machine, we have to perform your corrected WBC count. So again, you have your WBC count from the machine, multiply by 100, and divide the result by the total number of your NRBCs plus 100. So always remember that the NRBCs here, the number of NRBCs here will always be dependent on 100 WBCs counted, especially in the leukocyte differential count. Okay, so example, in your machine, the machine was able to register a result of 10,000 cells per microliters. Okay, what is the corrected WBC count? If you were able to count, for example lang, 22 NRBCs in the peripheral blood film. So what you need to do is you have to multiply your 10,000 cells per microliters to 100 and then divide it by 22 because that is the number of NRBCs you were able to count plus 100. So that is the corrected WBC count. Okay. So after this lecture, I want you to watch the video on how to stain your peripheral blood smear. And then right after that, kindly go back to our Google Meet so that if you have any questions regarding my discussion for today's lesson and activity, we can discuss it in the Meet. So thank you so much for listening.